Your exams don't do flap, neither do we. Straight talk without the sweet talk. All you have to do is listen, read, test, and destroy. Let's start this lecture now.
Let's continue with special characteristic organisms that are anaerobic, facultative anaerobic, intracellular, encapsulated and urease positive in exam testable detail. Anaerobic bacteria are organisms that grow best in environments with little to no oxygen because they lack adequate protective enzymes against reactive oxygen species. Specifically, many true anaerobes either completely lack or have very low levels of catalase and superoxide dismutase, enzymes that normally detoxify hydrogen peroxide and superoxide radicals generated during aerobic metabolism. As a result, exposure to oxygen leads to accumulation of toxic free radicals and rapid cellular damage, which explains their inability to survive in oxygen-rich environments. Clinically relevant anaerobes commonly tested by examiners include Clostridioides, Clostridium species, Bacteroides, Fusobacterium, and Actinomyces israelii. These organisms characteristically produce short-chain fatty acids during metabolism, which is responsible for the classic foul-smelling discharge associated with anaerobic infections, a frequent clinical clue embedded in vignettes. Their fermentative metabolism also generates gases such as carbon dioxide and hydrogen, leading to gas formation in tissues, which may be seen on imaging as gas bubbles in necrotic infections, abscesses, or clostridial myonecrosis. Anaerobes are notoriously difficult to culture because they require specialized oxygen-free conditions, a detail often tested when blood cultures remain negative despite clear clinical infection. Although anaerobes normally colonize the gastrointestinal tract and oral cavity as part of normal microbiota, they become pathogenic when displaced into sterile sites, such as aspiration of oral contents into the lungs causing aspiration pneumonia or spread into deep tissues following trauma. A critical pharmacologic concept repeatedly tested is that aminoglycoside antibiotics are ineffective against anaerobes because oxygen-dependent active transport is required for these drugs to enter bacterial cells. Without oxygen, the drug cannot reach its intracellular ribosomal target. Facultative anaerobes represent a metabolically flexible group of bacteria that can switch between aerobic respiration and anaerobic metabolism depending on oxygen availability. When oxygen is present, they use it as the terminal electron acceptor to efficiently generate ATP via oxidative phosphorylation. In hypoxic or anaerobic conditions, they shift to fermentation or other oxygen-independent pathways to survive. This adaptability explains why facultative anaerobes are among the most common human pathogens, capable of thriving in diverse tissues. High-yield examples include Streptococci, Staphylococci, and enteric gram-negative rods. Examiners frequently test this concept by describing infections occurring in both oxygen-rich environments, such as the bloodstream, and low-oxygen environments, such as abscesses, emphasizing why these organisms are so clinically versatile and prevalent. Intracellular bacteria are classified based on their dependence on host cells for survival and replication. Obligate intracellular organisms, such as Rickettsia, Chlamydia, and Coxiella, are entirely dependent on host-derived ATP and metabolic machinery, rendering them incapable of surviving outside host cells. Their intracellular lifestyle protects them from many immune defenses and certain antibiotics, which explains why infections with these organisms often require antibiotics capable of penetrating host cells. Exam questions commonly hint at this category by describing organisms that cannot be grown on standard culture media and instead require living cells or specialized systems. In contrast, facultative intracellular bacteria, including Salmonella, Neisseria, Brucella, Mycobacterium, Listeria, Francisella, Legionella, and Yersinia pestis, can survive both inside and outside host cells. Their ability to persist within macrophages allows them to evade immune killing and disseminate systemically, which is why these infections often present with prolonged fevers, granulomatous inflammation, or relapsing disease patterns. This intracellular survival strategy is a recurring test theme linked to chronic infection and immune evasion. Encapsulated bacteria possess an external polysaccharide capsule that serves as a major virulence factor by inhibiting phagocytosis. The capsule interferes with complement deposition and prevents effective recognition by neutrophils and macrophages. High-yield encapsulated organisms commonly tested include Pseudomonas aeruginosa, Streptococcus pneumoniae, Haemophilus influenza type B, Neisseria meningitidis, Escherichia coli, Salmonella, Klebsiella pneumoniae, and Group B Streptococcus. From an immunologic standpoint, capsular polysaccharides alone stimulate a T-cell independent immune response, which is weak and produces limited immunologic memory, particularly in young children. Conjugating these polysaccharides to a protein carrier converts the response to a T-cell dependent one, enhancing antibody production and long-term protection, a concept frequently embedded in vaccine-related questions. Clinically, these organisms are normally cleared by opsonization and splenic macrophages. Therefore, individuals without a functioning spleen have impaired clearance of encapsulated bacteria and face a markedly increased risk of overwhelming, rapidly progressive infections. Board-style questions often test this association by presenting severe sepsis or meningitis in asplenic patients and emphasizing the need for targeted immunization against these organisms. Urease-positive organisms possess the enzyme urease, which hydrolyzes urea into ammonia and carbon dioxide. The release of ammonia raises the local pH, creating an alkaline environment that promotes bacterial survival and alters host tissue defenses. High-yield urease-positive pathogens include Proteus, Streptococcus, Helicobacter pylori, Ureaplasma, Nocardia, Klebsiella, Staphylococcus epidermidis, and Staphylococcus saprophyticus. This biochemical activity has important clinical consequences. In the urinary tract, elevated pH favors precipitation of magnesium ammonium phosphate, leading to struvite stone formation, a classic association particularly emphasized with Proteus infections. Examiners frequently test this by linking recurrent urinary tract infections, alkaline urine, and large branching renal stones. In gastric infections, urease activity allows organisms like H. pylori to survive acidic environments by locally neutralizing stomach acid, contributing to chronic gastritis and ulcer disease.